So now this is what you call that. You can see that this is the updated version of fluid mosaic membrane model, very recently updated model of this fluid mosaic model, right? So you can see very beautifully, right? It is cut. The cellular inside the cell also has been shown here, right? Why that inside the cell is shown? You can see the yellow lines and the brown lines are what you call that. These are the cytoskeletal elements. That what I want to show you is actually you can see, right? If you look, follow my cursor. Now this is one of the integral protein. Now this is also integral protein, right? And then you can see, and all these are the integral protein can be completely span the bilayer. Then it is called transmembrane protein, or else they can be half immersed or embedded inside the bilayer. Understand? Unfortunately, you can see some of the proteins here, right? The peripheral proteins. So now these are the peripheral proteins here, right? And these are all these blue integral protein and things like that. You can, these are the integral proteins, right? Okay. And you can see the carbohydrate, this is the inside the cell, and this is the carbohydrate moiety. Now you can see all the carbohydrate moieties are attached to, now this is what you call glycoproteins. Now these glycoproteins are the one involving in the cell recognition. And then I told you the lipid draft certain phospholipids are isolated around and they surrounding the particular integral protein area. That is for a specific function. Maybe through that a particular lipid can be transported, specific function. So I mean exactly what I'm trying to say here, the phospholipid bilayer is not homogeneous, it's a heterogeneous. Even among the heterogeneous also, some of the phospholipid can be together. Understand? And then that is the structure is shown here. Okay, that's all about the fluid mosaic model. So what you have to do homework is basically why a bilayer or membrane, right, is asymmetric. How it is asymmetric? What are the phospholipids are present inside? What are the phospholipids are present outside? And then what is the role of peripheral protein? What is the role of what you call that integral protein? Why this is making a curve, right? That means membrane making a curve and forming a sac-like structure. And then you should know other aspect of something like that. For example, when a membrane is formed, right? When this asymmetry have some sort of what you call that functional purpose, right? That means the bilayer, asymmetric nature of the bilayer, right, related to the membrane function. Understand? So you must understand whether this protein can move, whether these lipids can move in between two monolayers, etc., etc. You have to find out why this is maintained, why this asymmetry is maintained, how this asymmetry is maintained. You have to find out. Okay, that's your homework. Anyway, with these things we have done on the day about the cholesterol and membrane function, transmembrane protein, everything has been done. And then we are here now, I think, isn't it? Right? So, and then now, for example, right, now these so-called, what do you call that, receptors, the role of membrane carbohydrates. The role of membrane carbohydrate is actually cell-cell recognitions. When you say cell-cell recognition, cells recognize each other by binding to surface molecules, often containing carbohydrate, right? This can be a glycolipid or this can be a glycoprotein, right? So most of the time, this carbohydrate is like a short chain, right? Short chain carbohydrate. Then carbohydrate lectures, Dr. Nisanshal, I will teach you. That is what we call that oligosaccharides. So the oligosaccharides are attached to the protein. And then you can see now the proteins are not branched in the cell membrane, but carbohydrates which are present in the cell membrane are branched also. And then how these branches take in place, Dr. Nisanshal, I will teach you. And then you can see glycolipids and glycoproteins. They are the one responsible for the cell-cell recognition. And then these glycolipids and glycoproteins are collectively called as glycocalyx, right? Glycocalyx vary among species, individual, and even cell type. Okay. So now you can simple example, you can see now this is the most of the time these glycoproteins act as what you call the receptors, right? And then you can see now HIV infection, right? For the HIV infection, there's a particular receptor is there, right? That is CCR5, right? And then CCR5 is the co-receptor. The receptor is CD4. That don't worry about the name, but there are these two are glycoproteins. And then you can see now they recognize a particular what do you call that protein on the surface of the HIV virus, isn't it? And then that is what is happening. The infection is taking place. The genome has been transferred into the what do you call that the virus particle genome has been transported into the host cell, right? So this is this is what you call that HIV infection, right? HIV cannot infect a cell lacking CCR5 on the surface as in resistant individual. Then you can see now this person is the first person infection is taking place, and the second person is infection is not taking place. So CCR5. So you can see person to person, cell to cell, this variation can happen. That is how the infection also. You can see some people COVID virus can easily infect. In some people, they say immunity is good and all this nonsense, right? They use what do you call Pani Bonava, that what do you call that various Ayurvedic medicine they are drinking. All these are useless. Simple, right? In the COVID also there is a protein called spike protein. And then in the lung parenchymal cells, we have our, what do you call that? There is a particular receptor. If that receptor is coming in, this spike exactly fitting to the spike protein. And then you can see none of the animals are infected. Only we are infected. Maybe that receptor molecule is not in the animals. That's why COVID is not infecting the, what do you call that, animals. And sometimes you can see youngsters is not affecting them. Maybe the receptor is not developed fully in young people. That's one of the reasons COVID is not infecting them, right? Understand? So this is what you call that cell-cell recognition. It can recognize the own cell or it can recognize a different cell. When it is recognizing a different cell, it is rejection. And then when it recognizes the same cell, they form the tissue. Okay, right. Now this is what you call that. Just now I explained there's a coronavirus infection. So when you have a coronavirus infection also, you can see now that is the lipid membrane of the virus, and then spike protein is there. Spike protein is come and bind to the particular receptor, and then there is another what do you call that protein is needed in the cell membrane. So this is, you can see now that ACE2, that is the receptor fitting to the what you call spike protein of the coronavirus, and then that causes the infection. This is one of the typical example. The role of glycoprotein or a role of carbohydrate is recognizing and serve as a receptor. So it can recognize a cell or it can recognize an antigen, etc., etc. Okay, right. So synthesis and sidedness of membrane have distinct inside and outside phases. Right, that is what I want to talk about, right? The asymmetrical distribution of protein, that is your tutorial. The asymmetrical distribution of proteins, lipid, and associated carbohydrate in the plasma membrane is determined when the membrane is built by the ER and Golgi apparatus. So this is the show line that you can see now what is happening. The ER, the protein is synthesized. Most of the transmembrane proteins and synthesized, the glycoproteins are synthesized in the ER, and then they will be packaged or packed in a vesicle, and then that vesicle travels, right? Travels to the, what do you call that, what do you call Golgi apparatus, and the Golgi apparatus is undergoes modification, and then after modification, what happens? This is again repackaged in a vesicle, and then either if it is a secreted protein means usually what happened, secreted out, And then if it is a transmembrane protein, it's a protein belongs to that cell membrane that will be inserted into the membrane. And then this is how the membrane proteins are synthesized. And then you can see the phospholipids lipids are synthesized in the smooth endoplasmic reticulum. And then you can see the proteins are synthesized in the rough endoplasmic reticulum. Understand? Okay. So now what happened when you come to this, the functions of transmembrane protein, right? The major function, the membrane transport is involved by what you call this transmembrane protein. So the transmembrane protein can be divided into two. One is called passive transport, other one is we call active transport, right? When you come to passive transport, we can divide into what you call that simple diffusion. That means they can diffuse through the bilayer, water, right? Non-polar compounds. Some of the small ions also can go through the what you call that bilayer. This is what you call simple diffusion. There is one thing called is the transport is another one is called assisted transport or facilitated transport. So facilitated diffusion. When you say facilitated diffusion, what you can see, can you hear me?
Sir.
Yes, sir.
Yes, sir.
So the proteins can act as what you call that okay, the proteins can act as channels or carrier proteins, right? At the same time, there's another type of transport is there that is
Excuse me,
called,
sir.
We can't see the screen, sir.
can you see now?
Yes, sir.
Okay. So the proteins, another type of transport is called active transport. The active transports are usually, we call it as pumps, and then co-transporters are there in active transport, right? And then the larger molecules are transported, we call bulk transport. The bulk transport is two types. One is called endocytosis, other one is called exocytosis, other one is called pinocytosis. Okay, so there are transport mechanism explained here. One is the small passive transport and active transport. Passive transport can be divided in simple diffusion and facilitated diffusion. And then simple diffusion, water, non-polar molecules can go through the lipid bilayer. And then facilitated diffusion, there are channels and permeases. Channels are called channel protein, permeases are called carrier protein. And in active transport, there are pumps and co-transporters involving. And then the larger molecules are transported as bulk. These are called endocytosis, exocytosis, and pinocytosis. I told you I am not going into the detail of what you call that of transport mechanism. You will be studying in physiology, but my duty is when I talk about the membrane, I have to talk. So I put everything in one picture, so you have a simple idea. So one is, I said, is what you call the simple diffusion, non-polar compounds, right? What happened? They always transported down the concentration gradient. When you say the down the concentration gradient, mean outside concentration is high, inside concentration is low. So they will be transported down the concentration gradient, right? That is what you call simple diffusion. And then when you come to facilitated diffusion, right? There you can see there can be a electrochemical gradient. There is a electrical gradient can be, or else there can be a chemical gradient can be take place. So this is what you call facilitated diffusion, right? So here also what happened, down the concentration gradient. That means outside the concentration is high, inside the concentration is low. And then here you can see there's an involvement of a carrier protein or channel protein, right? So that a protein, that depending on the that carrier protein is formed by what? The protein, not necessarily ion channel. It can be formed by a hydrophobic amino acids. So the hydrophobic region can be there, right? And then through that substances can go through, and that is what you call facilitated diffusion, we call it, right? And then when you come to this active transport, when you come to active transport, there can be two kind of active transport we have. One is called primary active transport and secondary active transport. So now you can, when you go primary active transport means, right, we are transporting a molecule against the concentration gradient. So when you transport a molecule against a concentration gradient, what we need is actually outside concentration is low and then inside concentration is high. Understand? So what is happening here? When you are transporting a molecule against the concentration gradient, you need energy. That energy is supplied from a primary source. The primary source of energy is ATP. So ATP is converted to ADP. Using that, what happened? That solid is transported. So here also there is a protein is involved, right? So primary active transport also carrier mediated. Understand? And then you can see another one is called secondary active transport. Secondary active transport is, right, one is, right, against the electrochemical gradient driven by ion moving down its gradient. One is transported down the gradient, other one is transported against the gradient, right? So now, for example, most of the time, right, the substance which is transported down the gradient is an ion, right? So the ion transported down the gradient, and then you can see the substance is transported against the, against the gradient, right? So outside is low, inside is high. So the dragging force for the substance to go against the concentration gradient is the ion transport. So this is what you call co-transport. This is what you call co-transport. Api udarana vashayen gattu. Hello. Carbohydrate kama kana. When you eat carbohydrate, what happens? The carbohydrate will be digested in the intestine, right? Digestunama, we get glucose. The glucose is absorbed in the intestinal mucosal cell, right? When the glucose is absorbed in the intestinal glucose, whatever, in the intestine, glucose concentration is low. The cell, the glucose concentration is high. So what will happen? Usually, the glucose will be transported against the concentration gradient. So for you need that, you need a force, right? I told you, usually ATP is given as an energy source, then that is called primary active transport. But what will happen with the glucose? Suppose we say instead of this S, I put glucose. Very carefully listen to me. Instead of this S, I put glucose, and instead of this iron, I put sodium. So in the intestine, what happened? Glucose will go in together with the sodium. You know that? So when the sodium athleteanama, glucose is athleteanama, this is what we call co-transport. So sodium goes down the gradient, right? And then glucose goes against the gradient. Then what happens? So this is what we call secondary active transport. The energy doesn't come directly from ATP. So what happened? Sodium athlete and what happened? Cell negative charge, so the negative charge will be lost. So we have to put the sodium out. So you put the sodium out using a pump, right? Pump means what? Primary active transport. You spending ATP, we put the sodium out, and then we put 3 sodium out, and 2 potassium in, because to balance the charges, and that is what we call sodium potassium pump. We call it sodium potassium pump. So you can see the sodium potassium, sodium Elliot and sodium ethyl. So in the primary active transport kara, we can put the sodium out. At the same time, what happened? The secondary active transport, sodium and glucose is coming in together. So the secondary active transport, indirectly you need to spend energy. And then the primary active transport, there's a direct ATP involvement. Okay, that is the difference between the primary active transport and secondary active transport. And then you come to the ion channel. Ion channels also down the electrochemical gradient. Usually the protein forms the ion channel, and then through that ion is transported. And then there are two kind of what do you call that? These ion channels. Opening and closure of ion channel depend on two things. One is ion, right? Ligand. Other one is an ion. So we call it as a ligand-gated ion channel or ion-gated ion channel, right? So ion-gated or ligand-gated ion channel kara, ions can be transported, right? That is what we call ion channel down the concentration gradient. And there is something called ionophore mediated, right? Most of the drugs, antibiotics, some of the antibiotics are delivered, right? Because that particle is called ionophore that can fit the ion, right? In the center of that ionophore, right? This ionophore is what we call that basically hydrophobic, that can easily go through the bilayer, and then inside the cell, the ion can be released. So all in all, this is all the transport mechanism in one slide that you will study in physiology. Simple diffusion, facilitated diffusion, primary active transport, secondary active transport, ion channel, and ionophore transport. Understand? So some of these usually show the saturation kinetics. You will go into detail in the physiology later on. Just I leave it here. Okay. Then what I said, something again called bulk transport. The bulk transport is usually, right? For example, there are several bulk transport is there. One is called endocytosis. Other one is called exocytosis, right? Other one is called Pacocytosis. Other one is called Pinocytosis. Other one is called macropinocytosis. Other one is called vesicle related secretion. Other one is called non-vesicle related secretion. But it is simple. What do you mean by this? Substances can come in, substances can go out in bulk. Understand? Yeah, earlier when we talk about the transport, small molecule transport, how it is happening, simple diffusion, facilitated diffusion, primary active transport, secondary active transport, ion mediated ion channels, and then ionophores. Here what happened? Molecules are transported in bulk. This is called bulky transport, bulk transport. So exocytosis, some of the substances we want to send it out, right? For example, right, we are taking a bacteria. A bacteria is, or virus is taken by what you call the cell, right? You can see the both side, there are receptors are there, and then the particle will come and bind to this receptor, right? So there is a vesicle form inside that is called endocytic vesicle or endosome. That endosome fused with the lysosome, right? Lysosome has digestive enzyme, so that will kill the particle, right? So pathogenic particle, whatever it is, and then excreted out. So excreted out means you can see that process is called exocytosis, right? And then you can see some of the substances can be taken as bulk, right? By engulfing these substances, right? So there are pits. In that pits you have what you call the receptors, and then you have what you call that there is a layer of red color protein. That is what clathrin coated pits we call it. In these clathrin coated pits only, you have these receptors. So what happened? The particles comes and bind, and then engulfing this particle, the particle can be taken inside. That is what you call endocytosis. When it is without receptor, non-receptor mediated endocytosis. When the receptor is present, that is what we call receptor mediated endocytosis. Okay, so once this comes in endosome, right, then there can be put it out by exocytosis. Clear? At the same time, right, so if a pathogenic virus or bacteria is taken by the cell and then the bacteria is killed inside the cell, is called Pacocytosis. Any nutrients or any other molecules can be useful for the cell, also can be taken in. That is what we call receptor mediated endocytosis. That is somewhere here you can see. And then anything unwanted can be secreted out. That process is exocytosis. And then anything synthesized also can be exported to other part of the body, like for example hormones. And then some proteins can be exported, right? So what is happening? They are synthesized and then they put it into a vesicles, right? And then the vesicle can be released. That is called vesicle related secretion. Or else what happens? Sometimes they can be secreted out without in a vesicle. That is called non-vesicle related secretion. Pinocytosis is when a substance is taken, right, in the form of a liquid. That is what we call pinocytosis. And now this is the drinking of the cell is called pinocytosis. Understand? Macropinocytosis, the larger molecules also can be taken. That is what we call macropinocytosis. So this is what we call bulk transport. Okay, fine. And you can see membrane protein has a peculiar function. That function is usually what happens? Forming junction. When the cells are associated with other cells, when the cells are formed, right, adjoining, but attached to another cells, usually what happens, the membrane proteins are involved, right? So you can see there are various type of what you call intercellular connection. You will study in anatomy. I'm not going into detail of it. And then it's called tight junction. And there's another one we call gap junction. And then another one we call desmosome, right? Another one we call adherence. And another one we call is hemidesmosome. So these are what we call intercellular connections. How a cell is connected to another cell via junctions. And then these junctions have special characteristics. Who are involving in this junction is usually the membrane proteins, right? So there are tight junction, gap junction. Tight junction, it won't allow anything to go, right? And then when you come to gap junction, it selectively allows the substances to go to the other cell. So you can see it's something like that: desmosome, adherence, and then hemidesmosome you have. So this is one of the typical, the enlarged version of the desmosome, gap junction, and tight junction. So you can see now, when you come to the tight junction, it is really tight, right? Doesn't allow anything to go in, right? And then what happens, you can see, right? When you come to gap junction, you can see the passage of ions and small molecules are allowed, right? You can see now there are two cells involved. In all these cases, you can see that there are two cells. Now, one plate is cell 1 cytosol, this side, and then other membrane is what? Cell 2 membrane, right? And then you can see intracellular filaments are there. It's called keratin filaments. Okay, that is what you call intermediate filament. We have done. We will be going to do, right? And then here also there. Then you can see there are proteins from that membrane. Cell number 1 membrane and cell number 2 membrane proteins do the connection. Understand? Now this is desmosome, we call cells attached to another cell. And now this is the gap junction, right? There is a protein formed by a protein barrel or protein channel formed by a protein called connection, right? Connexins and connection. Connection is a protein, and then that several syn is formed, there's called connection. And through that, small ions can go, large molecule cannot go. This is what you call gap junction. So desmosome, gap junction, nothing can go through that. That is what you call tight junction. For example, we have a blood-brain barrier, right? When you have blood-brain barrier, certain extent only substances can go, that means allowed to go to the brain, right? So we are not allowing all the substances cross the blood-brain barrier. So the blood-brain barrier have tight junction, right? It doesn't allow all the substances go through. Understand? So this is what we call our junction, cellular connections, we call it. Cells can be connected to another cell via the cell junction. Okay. And as I mentioned earlier, the cell-cell recognition is done by the carbohydrate moiety present in the, what do you call that, cell membrane, right? As you all know, this is what I want to show you, right? You can see the bilayer is there, right? So that lipid is there, and then you can see the in the phospholipid also carbohydrate can be attached, and then you can see the proteins are there, right? Interestingly, you can see now there are nice proteins are there, right? So we can, for example, there's a protein, the transmembrane proteins are there. Understand? When you have a transmembrane protein, what happens? Sugar residues are attached. These sugar residues are what you call not larger molecules. These are oligosaccharides. Only few numbers of carbohydrates. You all know that. And on the other day, Dr. Nisansala taught you all, right? What do you mean by monosaccharide? What do you mean by disaccharide? What do you mean by oligosaccharide? So here you can see oligosaccharide attached to the proteins, and then they involve in the cell-cell recognition. Now you can see now the bottom part is cytosolic part, and then the top part is basically, what do you call that, outside of the cell. So you can see now the transmembrane protein, there is absorbed glycoprotein. Another protein can be attached to the transmembrane protein. Funny, isn't it? That is, you can see that protein is coming from extracellular matrix. Now we say inside is intracellular, but is outside is extracellular, isn't it? The extracellular matrix is a tissue present outside the cell, and then that also has protein. That protein can be associated with transmembrane protein. And then you can see inside the cell also, in the cytosol area, there are proteins called cytoskeletal protein. That those also connected to this transmembrane. So you can see extracellular matrix is outside the cell. Those proteins also connected to this transmembrane protein. And then in the intracellular area, there are cytoskeletal protein that also connect the transmembrane protein. And you can see, so the transmembrane protein have some function other than what you call that transport. That is what you call cell-cell attachment, right? Cell-cell attachment and cell matrix attachment. Matrix can outside, extracellular matrix attachment. So these proteins are involved. That is what I wanted to do a homework. What are the functions of peripheral protein? I will be teaching in the tutorial, don't worry. And what are the functions of what? Integral protein. And now somebody can, looking at this picture, can ask a question. I want an intelligent student to ask a question. I am giving 2 minutes.
So why there's no any carbohydrates in inner membrane?
Yes. Can you tell me in louder, please?
Sir, why we can't see any carbohydrates in inner membrane?
That is your homework. Okay. That is your homework. Asymmetry. Okay. That is your homework. Why this asymmetrically distributed? Okay. Any other question?
Sir,
Yeah.
Glycoprotein and Proteoglycan are
Ah, very good. Who is this? Can I know the name?
Sharafa.
Sharafa. Okay, Sharafa, very good question you asked. That is the question I expected also, right? You can see, one is we call glycoprotein, other one is we call proteoglycan. Mokadda me venas. They know that. So, glycoprotein and proteoglycan. Wow. Right. So you can see one is at the end of the name, one is protein. Other one is end of the name, what is what? Glycan. Right. Glycan is carbohydrate. So if the carbohydrate component is greater than the protein component, then it is called proteoglycan. Understand? And if the protein composition or the percentage in the molecule is higher than the carbohydrate percentage, that is called glycoprotein. And right? That end in protein, that means protein is more, less carbohydrate, then it's called glycoprotein. And then you have carbohydrate and protein together, but carbohydrate is high, protein is less. Then we call it as what? Proteoglycan. So you can see on the surface of a cell membrane, you can see the beautiful layer, right? Glycoproteins are there, absorbed glycoprotein is there, and then carbohydrates are attached to protein, transmembrane proteins are there, and then you can see some of the protein are coming from what you call intracellular and then going to extracellular. These are called transmembrane proteoglycan. So outside the cell, there's a code, isn't it? අපි ඇඳුම් අදිනවා වගේ තමයි ඉතින් නේද? නැත්නම් මිනිකමුණෙන් එන්න නැතුව මේ ෂර්ට් එක දාගෙන යනවා වගේ, that there's a code. Right? That is what you call glycocalyx. This is the one responsible for when a cell recognizes another cell. This glycocalyx is important. Now what happened? Most of the time in the cell, we call it, that is called antigens are coming in. What is antigen? Simply a foreign body entering the body is called antigen. And then our immune system is getting alerted, isn't it? So when the immune system getting alerted means, what is immune system? Our B cells, T cells that you have done in your A level. So what happened? A pathogenic organism enter the cell, right? And then what happened? These carbohydrate moieties can serve as the receptors, right? Glycoprotein. That is what happened with the COVID virus, isn't it? We have unfortunately had a receptor in our lungs that is for the spike protein present in the COVID. So how do they make the vaccine now for the COVID? Either they send the spike protein attached to adenovirus. Actually, this COVID came from an adenovirus. Adenovirus is the one generally it forms the cold for us, isn't it? Then one of the, whether it is created virus or it is naturally formed virus, that is not our argument here, right? So what happened? They attach that protein, right? Spike protein to an adenovirus, which is not harmful, and then what send it to the body, and then our body will produce what? Antibodies for that. That is one way of doing antibodies. That is what you call Covishield, and then this what you call AstraZeneca, Covax, and then what do you call that? Sputnik, and then what do you call that? The Chinese one, Sinopharm. All these are that kind of vaccine. And then what happened? The Americans did differently. What they did, that mRNA, which is responsible for the synthesis of the spike protein, right? So the vaccine is only having the mRNA that will go, and then in the host cells machinery, it will produce that protein only. That protein, without virus, the protein is no harm, isn't it? What happened? That protein, antibodies will be produced. So that is what they say, that is very efficient. But that we can't keep it here in Sri Lanka because mRNA very unstable. So you have to keep that vaccine in -70. So imagine -70 freezer we have to have all over the Sri Lanka is very unlikely, isn't it? So what we wanted to have the other vaccines, and then for some time in a rich countries or developed countries that Pfizer and Moderna is possible, but in our country, not only it is costly, but the logistic problem will prevent us using those. But our government also trying to get something like that. Okay, anyway, that is out of context. So this is what you call glycocalyx. Okay. So once you know the glycocalyx, we go inside the cell now. All this time we are talking about what? Membrane, and then asymmetry and all blah blah. Now I am going to talk about the endoplasmic reticulum. So endoplasmic reticulum is one of the cellular organelle, right? And then you can see that the present in almost in all eukaryotic cell, ER is often occupies most of the cytoplasm. It is a network of membrane. Okay, so the most present manner of origin of the endoplasm is not definitely known. The most concrete hypothesis is that ER is budded off from the nuclear envelope, right? So you can see that the nuclear membrane and the nuclear envelope, the continuation of the nuclear envelope is our endoplasmic reticulum, right? That is what you call the rough endoplasmic reticulum. As you all know, the nuclear envelope, which is covering the what you call the genetic material in a eukaryotic cell, is we call nuclear envelope. In a prokaryotic cell, the nuclear envelope is not present. The continuation of the nuclear envelope is the network of membrane. That is what you call endoplasmic reticulum. Okay, right. Can you all hear me?
Yes, sir. Yes, sir.
Okay. Are you all following me?
Yes, sir.
Yes.
Am I going fast? Am I going slow?
No, sir.
Okay, right. Anyway, right. So ER appears to arise from the outer membrane of the nuclear envelope, folding, or from the plasma membrane by infolding, right? You can see now there are two theories postulated. Either ER came from the cell membrane, that is what you call cell membrane is infolding and forming the ER, or nuclear envelope outfolding and form the ER. The smooth ER seems to arise from rough endoplasmic reticulum, detachment of ribosomes. Right? So, smooth endoplasmic reticulum derived from rough endoplasmic reticulum when it's shedding out the ribosome. Right? That is what you call smooth endoplasmic. Now, there's something wrong with okay, can you see the screen now?
Yes, sir.
So you can look at this schematic diagram of endoplasmic reticulum, right? Okay. So what do you see that there is a area, we call it as what you call cisternae of rough endoplasmic reticulum, and then you can see the cytosol, and then on top of that you can see the small particles like that is what you call ribosome. So when you look at the endoplasmic reticulum in a electron microscope, that is the right hand side picture will be that. Okay. And then you can see the scanning of electron microscope of the rough endoplasmic reticulum. And then this is what you call the immunofluorescent staining. There's a nucleus, and then you can see the continuation of the nuclear envelope. You can see that ER is here and there, the ER is there, right? So the new immunofluorescent staining of the endoplasmic reticulum, and then so that the postulation inward what do you call that, folding of the plasma membrane or outward folding of the nuclear envelope form the endoplasmic reticulum. Okay, so now you can see the beautiful endoplasmic reticulum, isn't it? Okay, right. So now you can see the light blue color. The light blue color is actually rough endoplasmic reticulum. As I told you, you can see the nuclear envelope. It has a lot of pores in it. You can the nuclear envelope is decomposed in a way. It's surrounded by what? Proteins. The nuclear envelope Nuclear pore is made by what? Proteins. And then you can see the continuation, the dark color, dark blue color, purplish color. And then it forms stack-like membranes, isn't it? And that on top of that, you can see the ribosomes. That is what you call rough endoplasmic reticulum. And connected to that rough endoplasmic reticulum, you can have the smooth endoplasmic reticulum. Okay, all right, fine. Shall I go to the next slide now? So the ER is a three-dimensional network of intracellular network. Three type of element, right? One is called cisternae. Sometimes we call tubules. Sometimes we call vesicles. So what is the difference between these three? Cisternae means these are flattened, unbranched, sac-like element. They lie parallel to another. So these, I mean, three forms of rough endoplasmic reticulum. One is called cisternae is the one where there is an unbranched, sac-like element and parallel to each other. They bear the ribosome on the surface. That's why they look rough. It has a glycoprotein on the surface. It's called riboporin 1, riboporin 2. That bind to ribosome. So ribosome kila nikamara nikan attach වෙලා නැහැ. අර සමහර වෙලාවට අපි කියනවා අර when ladies ke dress වල සමහර වෙලාවට sequence දානවා මුදු දානවා ඒවා දාලා they will make it pattern no නිකන් අර paste කරලා නැහැ. අර so sequence is now in a sari or something, what you do is you stitch it. Here also that ribosome to go and get attached to the what you call endoplasmic reticulum, you have some protein should be there. That is what you call riboporin 1, riboporin 2. And these are MCQ point, you see. And then when the ribosome attached, what do you call that, to the endoplasmic reticulum, chumma nave, right? It is attached to what? With the protein. Okay, right. Then we go to these tubules. These are irregular branching elements. So cisternae is not branch. Tubules are branch network. And then they are often free of ribosome. So in the ER has three type of structure. One is called cisternae is the one major one that is having what you call that ribosome. But when you come to tubules doesn't have what? Ribosome. And then you can see vesicles. Whatever synthesized in the ribosome. It's a ER is actually a factory, isn't it? And the ribosome on the surface. So when you can see the central dogma, imagine the central dogma now. Imagine the central dogma. What the central dogma says? DNA produces mRNA. Why DNA produces mRNA? DNA cannot come. Why it cannot come? It is large. Right? So what the DNA has? Genetic material. What is genetic material? Genetic information. What is the genetic information it has? What protein has to be synthesized? When it has to be synthesized? So switch on, switch off, everything is happening. So the message has to be given to the protein synthesizing machine. The protein synthesizing machine is what? The ribosome present on top of the endoplasmic reticulum. So a smaller molecule imprints the message in the DNA. That is what you call transcription. And then this small mRNA comes out. And then that is what you call translocation. And then it comes and attached to the ribosome. When it attaches to the ribosome, what happens? tRNA molecule bring the amino acids and then synthesis of protein taking place, isn't it? And these proteins are inserted in the lumen of the ER. When it is inserted in the lumen of the ER and they are packaged and they have to be given an address now. Now, api envelope එකට දැම්මා card එක දැම්මා envelope එක වැහුවා කාටද යවන්න ඕන කියලා ලියන්න නැතුව මේ post box එකට දැම්මොත් මොකද වෙන්නේ? කොහටත් යන්නේ නැහැ. So same thing for the protein. If the protein is inside the ER is synthesized and packaged in what? Small vesicles and then sent to the Golgi apparatus. Golgi apparatus, what it does, it does certain modifications. That is what we call post-translational modification, like addition of what you call that carbohydrate and truncating certain part, etc., etc., etc. So this is what we call post-translational processing. and then tacking the protein. Tacking the protein is important. Protein tack. Tack means what? Where to go? The protein has to be sent out. If it is a secretory protein, it has to be sent out. If it is a membrane protein, it has to be inserted into the membrane. So whether it is a membrane protein or whether it is a secretory protein, this has to be addressed. Where to go? Understand? So vesicles. These are oval and rounded and vacuole-like elements. They also don't have ribosomes. So ribosomes are only present in the cisternate. Tubules doesn't have. Vesicle doesn't have. And what is happening? This is the protein synthesized, packaged from this vesicle, and then vesicles comes out and then transported to the Golgi Apparatus. Okay, right. Now, the functions of SCR, smooth endoplasmic reticulum, right? The smooth and what is the difference between the smooth endoplasmic reticulum and the what is the difference between the rough endoplasmic reticulum? You can imagine කරලා බලන්න. අර ගෑනලා මේ ගත්තොත් මූණ ටිකක් නිකන් ලස්සන වෙලා මේ ෆේෂියල් කරලා නිකන් ස්මූත් ඉතියි. අපේ මූණ දැක්කොත් නිකන් මේ තේරෙනවද? නිකන් රෆ් වෙලා ෂේප් ගත්ත නැතුව නිකන් සමහර වෙලාවට පිම්පල් තියෙනවා පුදුම කැතයි, right? So that is the difference, right? So endoplasmic reticulum and then smooth endoplasm, everything is smooth. No ribosome, right? And what is the function? If the rough endoplasmic reticulum is for protein synthesis, protein packaging, protein modification, and then sending the protein to the correct place, none. Lipid synthesis කරන්න කවුරු කෙනෙක් ඉන්න එපා. So the major function of what? Smooth endoplasmic reticulum is lipid synthesis, carbohydrate synthesis, some of the carbohydrates, detoxification. That is why you can see smooth endoplasmic reticulum is abundantly present in the liver. Why it is abundantly present in the liver? Liver is the major detoxifying organ in our body, isn't it? Why we have to have a liver, isn't it? Ape body illuwane Coca-Cola bonda කියලා නේද? The body never asked Coca-Cola. The body never asked Sprite. The body never asked apple juice. Body wanted what? Water. Why water? To keep the body rehydrated. But we drink all this nonsense, isn't it? Body want carbohydrate. Body want carbohydrate. Body doesn't want nasi goreng, doesn't it? So what happened? So unnecessarily we take drugs in a sense, medicine, and then we take a lot of uses in the form of colored drinks and things like that. So who will detoxify this liver? So one of the major functions of the smooth endoplasmic reticulum, I will teach you in the xenobiotic metabolism later on, is what you call that, this smooth endoplasmic reticulum. So the mammalian liver, skeletal muscle, kidney tubules, and steroid producing endocrine glands and gonad cells, very abundantly present, smooth endoplasmic. Synthesis of steroid hormones, right? Steroid hormones, the steroid hormones. What are the steroid hormones? කතා කරමු steroid hormone මොනවද තියෙන කියලා කතා කරමු. Progesterone, estrogen, right? Aldosterone, all these are steroids. Yeah. Testosterone, everything you know, right? Okay, fine. So these steroid hormones either synthesize in gonad, right? Testis synthesizes testosterone, ovary synthesizes estrogen, placenta synthesizes or progesterone, or what do you call that? Yellow body synthesizes progesterone, etc. Okay. So in this, you have a lot of smooth endoplasmic reticulum. It also synthesizes phospholipid on the balance. Cell which secrete this product. Our cell membrane ekata adala phospholipid also synthesized by phosphatidylserine, phosphatidylcholine, phosphatidylethanolamin, inositol triphosphate, all these synthesized where in the smooth endoplasmic reticulum. In testes, ovaries, and skin, oil glands have great deal of smooth endoplasmic reticulum. Okay, right, fine. Because of this steroid hormone synthesis. Okay, so that is all about endoplasmic reticulum. Right, Metma, are you there?
Yes, sir.
Metma, what is the subject? Sorry, what is the next class for you have?
Anatomy practical, sir.
Anatomy practical, right? Okay, fine. And then evening, what you have?
Mentoring classes, sir.
At what time you have starting it?
2 o'clock.
Okay. Do you have any objection to have a lecture at 4 o'clock? 1 hour? Are you really tired?
No, sir.
Huh?
sir.
Okay. Shall I make a link and send it to you to have lecture at 4 o'clock?
So there's
Okay, sir.
we have the mentoring class. So it will be like continued for more than 1 hour sometimes.
Your mentoring session at 2 o'clock, isn't it?
No, for us, Dr. Andra sent the link for 3 o'clock.
3 o'clock. So what about 4:30?
That will be alright.
Huh?
We'll inform him and come to the class.
Yeah, I will speak with your batch rep Mitma, and then I'll come back to you. Right? Okay. At the moment, I will continue the class. Right? Okay. So the detoxification is done by what? SCR, smooth endoplasmic reticulum, isn't it? So that is the SCR bring about detoxification in the liver. Right? It converts the harmful material like drugs, poison into harmless material. So the detoxification is carried out by the some of the oxygen transferring enzyme like oxygenases or cytochrome P450. Now, some of the drugs are detoxified and then made as water soluble. When it made water soluble only, it can be excreted in the kidney. So most of the time, the reaction taking place in the detoxification is oxygenases involved and hydroxylation is involved. Once it is P450, does that function. Cytochrome P450 in the liver. So make the compounds hydroxylated. When it hydroxylated, it will become water soluble. Okay, right. And then SCR, then some of the examples, smooth endoplasmic reticulum, right? You can see very specifically we said the function of the rough endoplasmic reticulum. The function of the smooth endoplasmic reticulum is basically, right, not very specific, very vast. Involved in lipid metabolism, involved in carbohydrate metabolism, and then you can see it is involved in detoxification. And not only that, it produces certain other cell organelles. Example, Golgi apparatus derived from smooth endoplasmic reticulum. Evage lysosome, vacuoles also derived from what? This endoplasmic reticulum, smooth endoplasmic reticulum. So, and not only that, it has receptors on cell membrane proteins and steroid metabolism. In muscle cell, it regulates calcium ion concentration. Then this is what important. That this is what I said. You dissect and study. Why muscle has lot of endoplasmic reticulum? Because they're regulating the calcium ion concentration. There's the involvement of smooth endoplasmic reticulum, and calcium is very important for muscle contraction, isn't it? Okay, right. So the smooth endoplasmic reticulum also contains the enzyme glucose 6-phosphatase, which convert glucose 6-phosphate to glucose, a step in gluconeogenesis that you will study later on about this. Actually, you can see now gluconeogenesis mean gluconeogenesis. Gluconeogenesis mean what? Synthesis of new glucose. So when it happens, synthesis of new glucose, for example, then samar velawata, then you are having a fight with your mother, and then you skip the meal tonight. I don't want to eat. So your mother is asking, hey, come and eat. So you said, no, I don't want. Why you don't want? You know, after your blood glucose is over, your liver has glycogen. That glycogen will be utilized to maintain the blood glucose overnight. Understand? So overnight fasting, you can very, what do you call that? Stubbornly say your mother, I don't want to eat. Because you know liver has glycogen. Your mother doesn't know. Because mother doesn't know biochemist unless she is a biochemist. Following morning, your mother is asking, Dua, Puta, can you come and eat? And then you still say, I don't want. Why you don't want? Because you know your liver can synthesize glucose. How your liver can synthesize glucose? Right? That is what you call gluconeogenesis. New glucose, gluconeogenesis. In that one process is there. Glucose 6-phosphate has to be converted to glucose. That enzyme glucose 6-phosphatase present in that. So you will say your mother, I don't want, because you can synthesize glucose for 2 days. After that, your mother will ask, can you come and eat? You have to go and eat because you don't have any other option. Otherwise, you will die. Right? So something like this. So smooth endoplasmic reticulum has a lot of functions like this. Okay. Then functions of RER, right? It has produces proteins. There are 2 types of proteins it can produce. One is what do you call that? Secretory protein and membrane protein, right? And then linking of sugars to the glycoprotein starts in the RER. So you add in the carbohydrate sugars to the protein is starting in the RER and ending in the Golgi apparatus, right? So RER produce enzyme precursors for the formation of lysosome by Golgi complex. Whatever the enzyme, very careful, whatever the enzyme present in the lysosome is produced in the RER and packaged. Okay. And smooth ER formation also from rough endoplasmic reticulum. Okay. Then protein synthesized in the ER. There are, I told you, there are 2 types of proteins can be synthesized. Protein synthesized on the ribosome attached to RER. Ribosome can be attached to RER also. Ribosome can be present as free ribosome also. So the ribosome attached to RER can produce secretory protein, integral membrane protein, the membrane protein, soluble proteins of ER, Golgi complex, lysosome, endosome, and this. And free ribosome, which is present in the cytosol, they can synthesize cytosolic protein, peripheral proteins, protein transported to nucleus, protein to be incorporated in the peroxisome and mitochondria. They are synthesized by the free ribosome. Understand? So this is what you call endoplasmic reticulum. Right? I think time is up, isn't it? I think we, what is the lecture? At what time your lecture?
10:30, sir.
10:30, but I had to give you half an hour break. No, it's not nice. Anyway, 10:30 to what time?
To 12:30, sir.
12:30.
Yes, sir.
Okay. Can you the who is having the Dr. Nandalal's this one? How many students?
About 20 students, sir.
Okay, so you are starting at 3 o'clock, right? And then you see at around 4 o'clock, if Dr. Nandalal not finished, you can tell him that we have another session with Dr. Jiva. Can you? Shall I put it at 4 o'clock? Is it okay? Does he do 2 hours?
Usually, yes.
Yes.
Yeah.
So what about 5 o'clock?
2, maybe 1.5.
Okay. So what about at 5 o'clock? If I put the lecture at 5 o'clock?
That's all right.
කතා කරන්නකෝ මොකක්ද යන්නේ කතා කරන්නත් ලැජ්ජයි.
That's all right.
If you want this be to completed, and then if you want me to do the cytoskeleton and then do the tutorial, please let me know, right? I can have extra hour for you. If you're free at home, right? You're not doing anything, isn't it? Follow, you know, together. Nani? Katha karanda, netma kauda me male batch rep is who is the male batch rep?
Cyan.
Who is this?
Hirun.
Huh? Hirun.
Yes, sir.
Hirun, katha karanda. Ogolida amaruda. If I have a lecture at 5 o'clock, if you don't want, it's okay, fine. I am fine with that. Let me know,
Let's do
right?
it
Okay.
at 5 o'clock.
Yes.
Let's do it at 5 o'clock.
Is it okay with others?
Yes, sir.
Yes, sir. Okay.
Yes, sir.
Okay.
Okay.
So we will do at 5 o'clock. I'll send the link to you. Okay.
Okay, sir.
Now I finish. Okay. Thanks.
Thank you very much.
Thank you.
Thank you very much, sir.
Thank you, sir. Thank you, sir.
Thank you, sir.
Sir.